student. In today's class we are going to study alternation in genetic makeup and its significance. Okay, here we are going to study the change in chromosome structure and chromosome number. So first we are going to see what is the chromosome. So here in the nucleus of each cell, the DNA molecule is packaged into a thread-like structure. This is called as a chromosome. Each chromosome is made up of DNA tightly coiled many times around proteins called as histones that supports its structure, and each chromosome has a constriction point is called as the centromere into two sections or arms. So here you can see the structure of the chromosome. So here you can see the double helix structure of DNA molecule, and this DNA molecule prepares the chromosome structure. And here this chromosome having the two arms, and the middle one, this one is the centromere, and it's having the P arm and Q arm, and so these are made from the DNA molecule and histone protein. Then next, the chromosomal mutation. So that means the mutation means the changes. So chromosome changes, how it will be takes place. It is an unpredictable change that occurs in the chromosome. In a chromosome, these changes are most often brought by problems that occurs during the cell division or by mutagens like chemicals, radiations, etc. Chromosome mutation can result in change in the number of chromosome in a cell or changes in the structure of chromosome. Then first we are going to study the change in the chromosome structure. So that is, change means the variation in chromosome structure. There are four common types of chromosomal mutation involving changes in the chromosome structure. First one is deletion, duplication, second one is duplication, third one is inversion, fourth one is translocation. So there are four common types of chromosomal variations are there. So one by one we'll see the variation in chromosome structure. First, here we can see the deletion and duplication. Deletion and duplication, these are the two types of structural mutation. This involves the change in the amount of DNA on a chromosome. So deletion and duplication involves the change in the amount of DNA on chromosome. The next one is the inversion. Inversion involves a change in the orientation of the chromosomal segment. And fourth one is translocation, and translocation involves a change in the location of the chromosomal segment. So one by one we'll see. First one is deletion. So here you can see the picture, the diagrammatic picture of the deletion. It is a chromosomal mutation in which a part of chromosome is missing or deleted. So here you can see the structure break in the chromosome. Here in the figure you can see the break in the chromosome. So it will get missing. Since the segment of chromosome is missing, so deletion mutation cannot be revert to the wild type. So here a centric chromosome, if the deletion involves the loss of the centromere. If it is involves in the center or the center part, centromere part, then what happened, the centromere will be lost here. So this is what the deletion. So here some human disorders are caused by deletions of chromosomal segments. So here Cri-du-chat is one of the human disorder caused by the heterozygous deletion. A segmental deletion in chromosome number 5 causes this syndrome. Children with this syndromes are severely mentally retarded. So here with this uh, deleted segments of the chromosome, can symptoms of the in the children, these children are mentally retarded. So they can be they can't be work properly with their brain. Many physical abnormalities occurs in this children. Cry with a sound like a meow of a cat. So they cry like a cat, About 1 out of 50,000 live birth has Cri-du-Chat syndrome. And then next, another example of chromosomal deletion is Prader-Willi syndrome, result due to deletion of long arm of chromosome number 15. Sucking reflex of the affected infants are weak, which results in a poor growth. But at the age of around 5 to 6, children with this syndrome becomes compulsive eater, producing obesity and related health problems. Reasons still unknown. About 1 to 1 in about 1 in 25,000 suffers from this syndrome, predominantly males. And then next one is duplication. A duplication is a chromosomal mutation that results when a segment of DNA breaks off and attached onto the homologous chromosome. So here you can see the duplication of this chromosomal segment takes place, and it it get breaks and attached to the homologous chromosome. There are different forms of chromosomal duplication or different tandem configuration. So first one is tandem, reverse tandem, and here you can see the terminal tandem. So we'll see when the order of the genes in the duplicated segments in the opposite of the order of the original mutation is a reverse tandem duplication. So here it is a reverse tandem duplication. Here you can see this reverse tandem duplication. Then next one, if duplicated segments are arranged in a tandem at the end of the chromosome, the mutation is then called as a terminal duplication. So here you can see this one is at the end of this chromosome arm, the duplication will take place. That segment is known as the terminal tandem duplication. The next, duplication can be caused phenotypic variation that might at first appear to be caused by a single gene mutation. The bar eye phenotype in Drosophila is a classic example. Instead of the normal oval eye shape, bar-eyed flies have a narrow slit-like eyes. So here you can see the Drosophila. So their eyes structure will be eye structure or shape will be changed. Then next, inversion. It is a chromosomal mutation that results when a segment of the chromosome is excited and then re integrated at the at an orientation 180 degree from the original orientation. There are two types of inversion. So first one is paracentric inversion and pericentric inversion. Typically genetic material is not lost when an inversion takes place. So first we'll see here the paracentric is there and even the pericentric is there. So paracentric, we'll see the first we'll see the paracentric one. If the centromere is not a part of the rearranged chromosome segment, it is a paracentric one. So instead of the centromere, another part of the chromosome segment takes place in inversion, then this part is known as what? The paracentric inversion. And then next is a pericentric. If if the centromere is a part of the inverted segment, it is described as a pericentric. If here the centromere is a part of inverted segment, then it is described as a pericentric inversion. Then the fourth one is the translocation. If a segment of chromosome breaks off and attached to a non-homologous chromosome, then the situation is called as a translocation. The translocation can be of three types: non-reciprocal, intrachromosomal translocation, when a chromosome segment changes position within the same chromosome. So this is known as non-reciprocal intrachromosomal translocation. Second one is non-reciprocal interchromosomal translocation, when the chromosome segment is referred from one chromosome to another and it was a one-way transfer. Then third one is reciprocal interchromosomal translocation, when a chromosome exchange occurs between both the chromosomes. So that is known as the reciprocal interchromosomal translocation. Then here you can see the non-reciprocal translocation, reciprocal translocation. Here you can see and how the translocation takes place in the chromosome. So next we'll see the variation in chromosome number. Numerical, it is also known as the numerical aberration in chromosome. Numerical aberration are mainly of two types, that is the euploidy and aneuploidy, which are again further divided. So here you can see the numerical aberrations. So these are first they are divided into two types, aneuploidy and euploidy. Then aneuploidy again divided into hyperploidy and hypoploidy. And so in hyperploidy there are trisomy, so 2n plus 1, tetrasomy, so there is 2n plus 2, hypoploidy, that there is a monosomy, that is 2n minus 1, and nullisomy, that is 2n minus 2. And in euploidy, monoploidy is there, n, diploidy 2n, polyploidy, that is the 3n. So we will see aneuploidy. First we will see the aneuploidy. Chromosomal mutation which results in the variation of individual chromosome number are example of aneuploidy. Changes in chromosome number can occur in both diploid and haploid organism. The non-disjunction of one or more chromosome during meiosis 1 or meiosis 2 typically is responsible for generating gametes with abnormal number of chromosomes. Then non-disjunction is the failure of homologous chromosome or sister chromatids to separate properly during the cell division. So here you can see here it is this is what the here you can see the first division, that is the non-disjunction. So this one is the normal disjunction. So here the chromosome gets separated in normal, and here in first division it doesn't divided, and here you can see so here you can see this picture. So here you can see the aneuploidy. Then next one is what the nullisomy and monosomy. So nullisomy, a nullisomic cell involves a loss of one homologous chromosome pair. So the cell is 2n minus 2. Then monosomy, that is the loss of single chromosome. The human monosomics are conceived but non-survive embryonic and fetal development, with the exception of Turner syndrome, that's it 45X. These are not tolerated in animals. The next, trisomy and tetrasomy, it involves a single extra chromosome, that is a single extra chromosome, that is 2n plus 1. In general, the effects of trisomy parallel monosomy, but the addition of an extra chromosome produces somewhat more variable individuals. The next one is the tetrasomy. A tetrasomic cell involves an extra pair of chromosome, that is there are 4 copies of one particular chromosome. The cell is 2n plus 2. Aneuploid condition in humans. So here you can see the aneuploid, that is the trisomy, means it's having the 21 chromosomes, autosomal condition. And here you can see this chart here. Here double X is there. Double X means the extra female chromosome, and here that means it will be the males. And here the syndrome which produce in the male, that is known as the Klinefelter syndromes, means the extra X chromosome present in the males. So that's why it is known as the Klinefelter syndrome. So in this children or in this males, sexual immaturity takes place, no sperms, and the breast swelling takes place in this male. And then next is here it is there X and double Y is there. So here the Y is double, so male characters is more in this. So there is a this this syndrome is known as the Jacobs, and this um, the the offsprings will be tall or the males will be the tall. And here the triple X, that means be that means the the offsprings or the individual is female, and it's having the triple X. The tall and thin means they it's having uh, characters, that is uh, tall and thin, and menstrual irregularity takes place in this female. X and 0, so yeah, it is females, and this type of syndrome is known as the Turner's syndrome. And in this the symptoms: short stature, webbed neck, sexually undeveloped. Females are developed in this sex chromosome. And then next we'll see, here you can see the Down syndrome, Patou syndrome, Edwards syndrome, and this one is the Turner syndromes. And then next we'll see the euploidy. Euploid organisms have varying numbers of complete haploid chromosomal sets. Monoploids and polyploids involves variation from the normal state in the number of complete sets of chromosome. Monoploid and polyploids are lethal in the most animal species but are less consequential in plant. Monoploidy: a monoploid individual has only one set of chromosome instead of the usual two sets. Some male wasps, ants and bees, for example, are monoploid because they develop from unfertilized egg. Cell of a monoploid individual are very useful for producing mutants because there is only one dose of each of the genes. Thus mutants can be isolated. Polyploidy: polyploidy is the chromosomal constitution of a cell or an organism that has three or more sets of chromosomes. Almost all animals and plants probably have some polyploid tissues. For example, the endosperm of the plant is triploid. The liver of the mammals and perhaps other vertebrates is polyploid. Plants that are completely polyploids include wheat, which is hexaploid, that means the 6n, and strawberry, which is octaploid, 8n. Some animal species such as the North American suckers of freshwater fish, salmon and some salamanders are polyploids. Two types of polyploids are encountered in the plants, so aneuploidy and alloploidy. So aneuploidy, in this all the sets of chromosome originate in the some species. The condition probably results from a defect in meiosis that leads to diploid or diploid gametes. The cultivated banana is an example of triploid polyploid plant. In general, the development of seedless fruit such as grapes and watermelons relies on odd number polyploid. The next, allopolyploid polyploidy. In this, the set of chromosome involves come from different through usually related species. This situation can arise if two different species interbreed to produce an organism with one haploid set of each parent's chromosome, and then both chromosome set doubles. A classic example of allopolyploidy resulted from crosses made between cabbages and radishes by Karpechenko in 1928. And thank you. This is about the alternation in chromosome, alternation in genetic makeup in chromosome. Thank you. If any doubts, you can ask.